All right, let's move on to objective 2. Describe the functions of saliva, stomach acid, pancreatic juice, and bile, and explain how the secretion of each of these substances is regulated. So let's start off with saliva secretion. So the taste and the texture and the smell of our food will stimulate mechanoreceptors, chemoreceptors in the mouth, and that is going to relay signals to the salivary centers in the medulla and the brainstem, which then send signals via the autonomic nervous system that begin that long reflex pathway that we just described. So sight of food, smell of food is what stimulates saliva and kind of gets these initial part of the breakdown of food started. So we have these inputs that are coming from our head. They're sent to the brainstem. These get sent to the cerebral cortex, and then the parasympathetic and sympathetic activation is what creates the saliva. So if we're having a meal, we're going to have the parasympathetic system involved. That's going to be more of a watery type of saliva. Whereas if we're not having a meal, the sympathetic activation will produce a more thick, a more viscous, protein-rich saliva. So we're always producing saliva, but the makeup of the saliva, the contents of the saliva are going to be slightly different depending on the inputs to the medulla or the inputs to the brain. Okay, and as you would imagine, a more watery saliva is going to be more efficient at lubricating the oral cavity, at diluting the contents of the oral cavity, of allowing that food to begin moving through the GI. Whereas the sympathetic system gives us a more rich, thicker, viscous saliva, which doesn't move around, is not as slick as this more watery saliva. Let's talk about acid secretion in the stomach. And we're going to look at the exact mechanism by which acid is secreted. So first off, we're going to remind ourselves that this is a parietal cell. Remember, the gastric glands of the stomach have these gastric pits, and lining these pits, we have several types of cells. We had gastric, chief cells, having a brain fart here. We have chief cells, we have parietal cells, we have G cells, and we have those neck cells. So we said that the chief cells secreted pepsinogen. The parietal cells, which we're talking about here, they secreted acid as well as intrinsic factor. The G cells secreted gastrin, which we're going to talk about later on, and then the neck cells secreted mucus. So make sure you're reminding yourselves of the makeup of the stomach, of the cells that are found in the stomach. So here we're looking specifically at a parietal cell. And this is the mechanism by which a parietal cell secretes acid onto the lumen of the stomach. So it's a similar mechanism that we used in the renal tubules, where we have carbon dioxide and water being combined via the enzyme carbonic anhydrase to give us carbonic acid. That carbonic acid can then be dissociated into hydrogen ions and bicarbonate. Across the basolateral surface, we're going to exchange bicarbonate for chloride. Again, a very similar mechanism to the way that bicarb is reabsorbed in the renal tubules. And when that chloride comes into the cell, it's going to make its way to the apical surface, and it's going to be secreted passively across the apical surface. So that's going to be the chloride component to this hydrochloric acid. Now, the hydrogen ion component, the hydrogen ions that are dissociated from this carbonic acid, are exchanged here via this proton pump. So this is a proton pump, and it is actively moving potassium into the cell and hydrogen ions out of the cell by liberating ATP and using that energy to move both of these ions. And so that is going to be how hydrogen ions move across the apical surface of this parietal cell. So if we combine that hydrogen ion with the chloride that is being passively moved across here as well, that gives us the hydrochloric acid that makes the acidity of the stomach lumen. That is basically how hydrochloric acid is secreted. Okay. So let's talk about the stimuli for acid secretion. So acid is going to be secreted in response to the parasympathetic nervous system. in response to gastrin secretion from the G cells, as well as in response to histamine that's coming as a paracrine stimulus from cells as well. Whereas pepsinogen secretion is going to be regulated in sync with acid secretion. So it's also going to have the same three stimuli: the parasympathetic system, gastrin, and histamine. And let's take a step here, let's take a second here and remind ourselves that pepsinogen is secreted from chief cells, acid is secreted from parietal cells, but in order to activate pepsinogen to pepsin, we need that acid that's coming from the parietal cells. So they're going to have, again, the synergistic relationship. They're bringing about the same response because the acid that is secreted is needed to activate this pepsinogen to its active form, pepsin, so it can begin to work on proteins in the stomach. Okay. Let's look at how acid and pepsinogen are influenced by the cephalic phase of regulation here. So the stimuli for acid secretion and pepsinogen are going to be the sight of food, the taste of food, the smell of food, the process of masticating or chewing, and then the process of swallowing. All of this will activate the parasympathetic system, again, via that long reflex pathway up to the central nervous system, down on the parasympathetic fibers, and that will in turn stimulate gastrin secretion from the stomach, from the G cells. So here's a little pathway or a little flow chart that kind of simplifies this. We've got the initial stimuli here, the sight, smell, taste, chewing, and swallowing of food. That goes up to the central nervous system. That is processed as a parasympathetic signal. That parasympathetic signal responds onto the G cells to increase gastrin secretion. That's going to increase the amount of gastrin in our plasma, but then it's also going to have a signal onto the parietal and chief cells. And that's going to stimulate both the parietal and the chief cells to secrete acid and pepsinogen. Acid coming from the parietal cells, pepsinogen coming from the chief cells. And so both of these are going to be secreted together, and that acid is going to activate that pepsinogen into pepsin. So this is how all of that is kind of regulated, coming from the initial sight of food and leading on to the secretion of acid in the stomach and the beginning of the breakdown of protein in the stomach via pepsin. Now, as far as the gastric phase of regulation, it's kind of the same thing from here on, but just that now we're incorporating both the short and long reflex because we're now in the stomach. So the stimuli of gastric secretion, that's going to be the presence of protein in our meal, or peptides or amino acids, as well as the distension of the stomach. So these are going to be the stimuli for the gastric phase of regulation, and that is going to elicit the short and long reflex pathways, which are going to bring about the exact same responses. That is gastric acid secretion and pepsinogen secretion as well. So here's another flow chart that looks at the gastric phase of regulation. So the presence of proteins, the presence of amino acids, the presence of peptides in the stomach, as well as the physical distension, the physical stretching of the stomach, will bring about two responses. The chemoreceptors that detect this are going to directly elicit the short reflex pathway, and then the central nervous system is going to be elicited as a part of the long reflex pathway. So both pathways are engaged, and the responses are going to be the G cells secreting gastrin, and then the parietal and chief cells secreting acid and pepsinogen, which is converted into pepsin. So the same responses, but the pathway is now incorporating the short reflex pathway because we're now in the stomach, and the stimuli now is the presence of these things physically located in the stomach, and not things like the sight or smell, which are located in the head. Okay, let's talk about the inhibition of secretion. So how do we kind of cut this off, right? Remember that when we stop eating food, we need to cease these signals. Let's talk about how that is brought about. So the gastric phase is inhibited when food exits the stomach. And so the increased acidity also inhibits gastrin release. So they're kind of two stimuli here. The movement of food through the pyloric sphincter down into the small intestines, as well as excess acidity. So if the stomach becomes increasingly acidic, that is going to be kind of a negative feedback signal to shut off that acid secretion. In terms of the intestinal phase, the movement of food down into the duodenum is going to start inhibiting the secretion, as well as excess or increased osmolarity, excess fats and acid, or excess distension. So once again, if we start to see too much of these things, there's going to be a negative feedback onto the duodenum to shut off these secretions, okay? And this is going to elicit the long and short reflex by inhibiting acid secretion and inhibiting pepsinogen secretion to shut off the acid production and the breakdown of proteins. Okay, let's talk about the secretion of pancreatic juices, those enzymatic rich fluid coming from the pancreas, and bile. So the acinar cells, they have a small volume of primary secretions, which is mostly comprised of water, electrolytes, and those digestive enzymes such as trypsin, chymotrypsin, etc. And then the duct cells have a large volume that has mainly bicarbonate-rich secretion. So these are the areas that have, of the pancreas, that have two different types of secretions that are being released onto the duodenum. So there are certain hormones that are going to influence the pancreas and the gallbladder to either promote secretion or to inhibit secretion into the duodenum. So first off, the stimulants are going to be enzymes and bicarbonate that is coming in the duodenum. The composition here is going to be vary depending on the contents of the stomach. So as we saw here, if the contents of the stomach are not including proteins, not including fats, we're going to have more of a watery component to the secretions. Whereas if the contents are including lots of fats, lots of proteins, lots of acid as well, and a larger volume of secretion, we're going to primarily elicit the ductal cells to release their secretion, which is going to be more of a bicarbonate-rich fluid to help neutralize that chyme that's coming in. So the primary stimulant is going to be dependent on what the makeup of the food that's coming through looks like. If it has lots of protein, lots of fat, it's going to have one type of secretion. If it has a little bit of protein or less fat, it's going to have a different type of secretion. So this brings us to speaking about two hormones that influence the acinar cells and the duct cells, respectively. So CCK stimulates the acinar cells to secrete their enzymes. This is going to be a small stimulant for bicarbonate release, and CCK is also going to potentiate the effect of secretin, whereas secretin is going to be stimulating release from the duct cells. This is going to be a larger amount of bicarbonate fluid, and this is also going to potentiate CCK to release its contents as well. So let's kind of back up here and kind of break this down a little bit. We have CCK, which is that cholecystokinin. We have secretin. Both of these hormones are kind of bringing about the same response, but they're doing it in a different way. CCK is eliciting the acinar cells. Secretin is eliciting the duct cells. So they're going to be sending off two different types of secretions into the duodenum. One is going to be mainly enzymes, which is coming from the acinar cells by CCK, and one is going to be primarily bicarbonate, which is coming from this, the duct cells stimulated by secretin. But while they both have their separate roles, they're both going to be potentiating each other. So while CCK is primarily stimulating enzyme release from the acinar cells, it is also going to be potentiating secretin to secrete bicarbonate-rich fluid. And the reverse is true. While secretin is primarily stimulating the duct cells to secrete a bicarbonate-rich fluid, it is also going to be potentiating CCK to secrete that enzymatic rich fluid. So while they have their own kind of pathway, they're also stimulating each other to bring about their own respective responses. Let's look at the stimuli for these two hormones. So the stimuli for CCK is going to be fat and amino acids, whereas the stimuli for secretin is going to be acidity in the duodenum. And remember, CCK is going to elicit the acinar cells. Secretin is going to elicit the duct cells. So the makeup of those secretions are going to be slightly different. All right, so here is a flow chart that kind of hopefully simplifies that. We have increased acid in the duodenum. That is going to be detected by the chemoreceptors. They're going to elicit both of our pathways. Remember, we're further down the GI system, so now we're eliciting both the long and short reflex pathways. That is going to influence the endocrine cells of the GI, of the duodenum here, to increase their secretin secretion. And the secretin secretion is going to have an influence on the duct cells to secrete a bicarbonate-rich fluid. And the purpose of that bicarbonate-rich fluid is to neutralize the acidity, which was the original stimulus to begin with. So then that will have a negative feedback loop onto the contents of the duodenum, and that will neutralize the chyme. The presence of this bicarbonate fluid will neutralize that acidity of the chyme. Let's see if we have, okay. So let's talk about the pancreatic juice and bile now. So the regulation of bile in the duodenum involves secretin and CCK as well. Secretin is going to stimulate bile secretion from the liver. CCK is going to stimulate the contractions of the gallbladder, as well as the relaxation of the sphincter of Oddi. And if you remember, the sphincter of Oddi is that sphincter that's guarding the opening of both the pancreatic duct, and the common bile duct, which are opening into the duodenum. So the presence of secretin will stimulate bile from the liver. The presence of CCK will stimulate the contractions of the gallbladder and the relaxation or opening up of that sphincter. Okay, so here we can see the two different roles of CCK and secretin, and how they're kind of trying to bring about a similar effect. So we looked at this role, the presence of acid influencing the plasma concentration of secretin, and secretin having that influence of bile coming from the liver. And that bile is going to emulsify fats down here in the duodenum. And then the presence of proteins, the presence of fats, as well as the distension here in the small intestine is going to influence the plasma CCK concentration to rise. That is going to increase the contractions of the gallbladder and the relaxation or opening of the sphincter, which will also result in more bile flowing into the duodenum, which will also work to emulsify those fats and neutralize the acid. Okay, and so these two responses will eventually turn off the initial stimuli, which were the presence of acid and the distension and the presence of fats and proteins. Okay, let me pause here a little bit and see if there are any questions. Okay, now there are two sets of checkpoint questions that I want us to go over, but I just realized that they actually have responses. So we'll look at them later on, okay? Okay, let's continue on here. So let's look at objective 3. Define peristalsis and segmentation and describe the role of each in digestion. So we talked quite extensively about secretion, about the hormones that influence secretion, about the neural pathways that influence and regulate secretion. Now we're going to look at motility, the actual movement of the wall of the GI tract. Now this is primarily involving the muscularis externa, right? If you remember those layers of the GI wall that we drew last time, the thicker outer smooth muscle layer, which is called the muscularis externa, is the primary area that is contracting to bring about the movement of the GI tract. Now, the function of this is to mix, to mulch, to kind of propel all of these contents together, as well as to get them evenly coating the wall of the GI so that they can be absorbed appropriately. Okay. So we have these rhythmic waves of contraction of this smooth muscle that bring about peristalsis and segmentation. Now, peristalsis is the intermittent squeezing of different parts of the GI. And so I like to do this kind of mechanism. This is the best way to describe peristalsis. So one portion of the GI squeezes, the other portion of the GI squeezes, and then that also involves segmentation. That means that food is going to move from one area to the other area, but it's going to actually be moving back and forth. That's what segmentation is kind of alluding to. So when we squeeze that food down into this area, and then we squeeze this area, it's going to kind of squeeze back up, and that's what actually creates this mixing effect of the contents of the GI. So peristalsis is propelling food forward, and segmentation is really creating that back and forth that really kind of mixes everything together. So let's look at peristalsis more closely here. So peristalsis involves the layers of the GI. So if you remember that smooth muscle layer, the muscularis externa, it had two different orientation of the fibers. There were some circular fibers that changed the diameter of the tube, and then there were some longitudinal fibers that changed the length of the tube. And so during peristalsis, we have the circular muscle contracting and the longitudinal muscle relaxing. And that is going to decrease the diameter of the tube, effectively pinching off a portion of the tube. That's going to be followed by the distal segment, which has the circular muscles relaxing and the longitudinal muscles contracting, which will allow food to move down into the next segment, right? That is going to increase the diameter. So one portion of the tube is experiencing a decrease in the diameter, and one portion of the tube is experiencing an increase in the diameter, and that is what pushes food along the GI, propelling it from one area to the other. Let's look a little closer at segmentation. So segmentation is this motility where we have, again, because of the fact that we have two different types of fibers here, there are circular components, or the circular portions of the wall are going to contract, but then when they are relaxed, they're actually going to squeeze some portions of that segment, squeeze the content here back into the previous segment. Okay, so as I squeeze the proximal segment and I relax the distal segment, and then I squeeze the distal segment and relax the proximal segment, that is going to create the back and forth movement of food. So some of this food we can see here is going to start moving backwards, and by the time we're done, all of it is going to be well mixed as it moves back and forth, moving down the tube. So both of these processes, peristalsis together with segmentation, are required to move food along in one direction for the most part, but also create this temporary back and forth that effectively mixes up the contents really well and exposes all of the food contents to the wall of the GI, mixing that kind